Welcome to Biology Made Easy. We are still solving genetic questions. Today is on dihybrid crosses for A-levels or AS students. Welcome. The question is, a homozygous purple-flowered short-stemmed plant was crossed with a homozygous red-flowered long-stemmed plant, and the F1 phenotypes had purple flowers and short stems. When the F1 generation was test crossed with double homozygous recessive plants, the following progeny were produced: 52 purple flower short stem, 47 purple flower long stem, 49 red flower short stem, 45 red flower long stem. Questions: A. Explain the results fully. B. State the expected numbers of the different types of the progeny. C. State 4 reasons why the observed numbers are different from the expected numbers in question B. Now, this question is going to be broken down so that we can easily explain. A homozygous purple flowered, it means we are dealing with a purple flower. You write it down. Short stemmed, you write your short stem around it, was crossed with a homozygous red flowered long stemmed. So this was crossed, that's the cross, with red flowered long stem. The F1 phenotypes, so we write our F1 here, had purple flowers short stems. You see, so the F1 purple flowered short stem. When you get here, this and this cross to give this, then you should be able to write the genotypes. Because if purple flowered short stem cross red flowers long stem, and you can get purple flowered, it means purple is dominant. So we give the purple A. And it also means short stem is also dominant. So we give the short stem allele B. Because it's a homozygote, it means this is it. Here, this is the genotype, AABB. That's the genotype. It's homozygous. And it was crossed with a long-stemmed, red long stem. So this will be the genotype here. Small b. From this information, purple short is dominant over red long. So that is the genotypes we have here. And they said when the F1, this is the F1, we got purple flower shorts. That is the F1. When the F1 generation was test crossed with double homozygous recessive plants, right? So the F1, this is the F1, this is the F1, crossed with double homozygous recessive plants. And this type of cross is a test cross where the heterozygote F1 is crossed with a double recessive. That is a test cross for dihybrid inheritance. And when this cross was done, these were the progeny: 52 purple flower short stem, 47 purple flower long stem, 49 red flower short stem, 45 red flower long stem. This was the progeny. And we are supposed to explain the results fully. So this is how we explain our results. Purple flower short stem, crossed with red flower long stem. We have certain the genotypes from our analysis. And we have the F1. F1 is purple flower short stem, and the F1 is a heterozygote. And this heterozygote, F1, is test crossed with a double recessive. So here's our F1 test crossed. Here's our gamete. So if we decide this is the male gamete, we put the male gamete here, our Punnett square. We put the female gametes on this line, our Punnett square. This individual is purple short. This individual is purple long. This individual is red short stemmed. This individual is red long stem. And the numbers have been given here. So we brought the numbers here. This ratio is approximately 1:1:1:1. So this is how we explained, and we say that genes are not linked. They are on different chromosomes. That is how come we have this ratio 1:1:1:1 for a dihybrid heterozygote F1 test cross. So we have explained the results here. And then B takes the expected numbers of the different types of progeny. The total numbers is 193. If you put all these together, they were 193. And we have 1:4, 1:4, 1:4. So 193 divided by 4, each progeny should have been around 48 individuals. So this should have been 48, 48, 48, 48, approximately 48. But it's 52 and the least is 45. Alright, so the expected numbers of the progeny is that each of the progeny should have been around 48. But the observed numbers are these. So question C is asking us to state 4 reasons why the observed numbers, that is these, are different from the expected numbers of approximately 48 each. And these are the reasons. Fertilization is a random process, and then not all zygotes or embryos mature to be counted. Then sample sizes are small. If the sample sizes of the experiment is big, we can near the 1:1:1 very closely. Then other genetic interactions like linkage or epistasis come into play. For instance, you cross an F1 heterozygote with a double recessive and you get the ratio 1:1:1:1, then there is no linkage. But in our next question, we will see that linkage occurred, and so the ratios are very far away from 1:1:1:1. So this is a dihybrid cross. Thank you very much. Please like and also subscribe. Thank you and goodbye.